Okay. Hello, everybody. Welcome to today's excellence talk of the Cyprus Biological Society. We are starting our 10th season this year, and we are very happy to have as our first speaker distinguished guest, Dr. Costas A, who's also from Cyprus. Maybe his surname is a giveaway. And he will be talking about targeting lipid metabolism in pancreatic cancer. So just before I give the floor to Dr. Lyssiotis, I will just go through a brief introduction to his research. So please bear with me for 1 second. So Dr. Lyssiotis obtained his bachelor degree in chemistry and biochemistry from the University of Michigan in 2004, his PhD in chemical biology from the Scripps Research Institute in 2010, and then he completed postdoctoral training in the laboratory of Louis Candley at Harvard and Cornell. In 2015, he joined the faculty at the University of Michigan, where he is now the Maisel and Dode Professor of Oncology and Professor in the Departments of Physiology and Medicine. He leads the Tumor and Immune Metabolism Program, and he is the co-director of the Roger and Blondie Pancreatic Cancer Center at Michigan Medicine. Dr. Lyssiotis is also passionate about science education and training the future generation of scientists. He is the director of the graduate program in cancer biology at the University of Michigan, and in this role, he is an active lecturer and participant in graduate education. Additionally, he leads a large and highly diverse research lab that studies the biochemical pathways and metabolic requirements that enable tumor survival and growth. This work spans the areas of cancer metabolism, the tumor microenvironment, and immunometabolism, using and developing protocols in mass spectrometry-based metabolomics. Ultimately, his group aims to transition new information about these processes into targeted therapies for cancer and other diseases. Dr. Lyssiotis has published over 225 peer-reviewed research papers, and he's recognized among the top 1% highly cited scientists in sciences. He has also been recognized as the next generation cancer scientist by Trends in Cancer Biology as an MBioFAR awardee for excellence in biomedical research, and he has been the recipient of several Junior Scholar Awards, including being named a Lefkowsky Scholar, Kimmel Scholar, and Damon Runyon Dale Frey Breakthrough Scientists. So I will now give the floor to Dr. Lyssiotis, and if just a note, if any of you has any question, he or she can note it down at the Q&A section, and we can address it at the end of the lecture. And Dr. Lyssiotis, I want to welcome you in our 10th season of Excellence Lectures. Thank you for being with us today, and the floor is yours.
Thank you so much for that extremely kind introduction, Maria. And let me see. I've been giving talks and participating in academic medicine for 20 years, and that's the first time someone's pronounced my name right. So that's a unique pleasure.
Greek background.
Yeah, that does help. Cypriot specifically, background. Well, I don't know where everyone's coming from today. And so good morning, good afternoon, wherever you are. As Maria noted, if you jot down your questions, we can certainly cover those at the end, but I also gave her full leeway to interrupt me at any point if the question that is being asked feels like it would provide clarity. I run a cancer research lab, and we work on cancer biochemistry. And so those are the topics I'm going to be discussing at large. I will try to provide both the necessary simplified background context for people who don't work in the area without belaboring it for people who do work in the area. But if you do have questions and need additional clarity, I would love to do that. The whole point of us being here together is that I can relay some new information. So I want that to be received. And today I'll be talking about some newer work from our lab regarding the targeting of lipid metabolism in pancreatic cancer. Biology, we have come to appreciate over the course of the past several decades, in fact, that cancer cells in a tumor do not exist in isolation. that they are part of a complex ecosystem where the cancer cells exist and interact with a variety of other cell types, some of which are listed here and include a variety of immune populations, fibroblasts, complex mixtures of extracellular matrix, so on and so forth. And these cells give the tumor a variety of unique properties that enable the tumor to do things tumors do, like grow and be resistant to therapy and avoid the immune system. Well, one of the features that we observe across all solid tumor types is that they have disrupted metabolism. Or more specifically, what I mean by that is the cancer cells do not have across the cancer, equal access to things like nutrients and oxygen. Wastes will accumulate in some places. Changes in pH, changes in tissue architecture, and that impacts what the cell has to eat, the building blocks necessary for proliferation. And if you're a cancer cell, the job is to proliferate. So the changes in access to metabolic fuels and wastes and so forth are typically manifest by leaky or insufficient vascularization of the tissue. And what this all together means is that cancer cells are under complex metabolic challenges. And what my lab is interested in is understanding how the cancer cells rewire their metabolism to get the foods they need in order to grow as tumors. And we work, most of this work is done in a disease called pancreatic cancer. The major type of pancreatic cancer is called pancreatic ductal adenocarcinoma. It also goes by the acronym PDAC. And PDAC is it's the worst major cancer type in terms of survival statistics. So most patients diagnosed have metastatic disease, and we literally have no effective treatments for metastatic disease. There are some really exciting things coming through the clinical trial pipeline. I will touch on those later in the talk. But at current standard of care across the world, we have no way to treat this disease. The 5-year survival of patients with metastatic disease is 3%, and most patients present with metastatic disease. So it is horrible, and we need to think about, and we need to come up with better strategies to treat this disease. So my group has approached this from the perspective of metabolism, based in part on the fact that this is not an area that prior to the last, say, 10 years and the work that my group's been doing among others, it's not an area that's had previously experienced a lot of activity. And secondarily, pancreatic tumors are really a prototype for a number of those features I told you on the past slide relating to the sort of interactions within the tumor microenvironment and the limited availability to nutrients, and I'll expound upon that a little more here. So this is a typical histological image of a pancreatic tumor. You don't need to be a pathologist to appreciate I've done the heavy lifting here for you. So what I've circled in this line and labeled, this is the neoplastic epithelium, or these are the cancer cells. So what is all of this other junk around here? This is the tumor microenvironment. Pancreatic tumors are somewhat unique in that they can exist with as little as 10% cancer cells. In other words, if you have a tumor, 90% of that can be not cancer. It can be other stuff driven by that 10% of the cancer cells who are doing the orchestrating. So what is all that other stuff? It's fibroblasts and macrophages and some other cell types as well. And what they collectively do is lead to this really intense pressure. Like literally, if you touch these tumors, they're dense, they're hard like a rock. And what that means is that under these circumstances, the vasculature collapses. The tumors do not have access to the bloodstream. That does pose challenges, we think, to the delivery of drugs, but it also poses real challenges to how they're getting their nutrients, how they're getting their oxygen. And we know that these are important things that cells need to grow to make more cells. So that's been the heart of the question. How are the cancer cells rewiring their metabolism? And this seems to be a feature that's unique to pancreatic cancer relative to normal host physiology. And so can we now, knowing that, exploit that as a means to develop better therapies? So major questions in my group. This is the first. I just laid that out, sort of the context for that. As I'll talk a little bit more about in the next slide, there is one of the ways that they get nutrients is through cooperation with other cells in the microenvironment. And we call this cooperation. So an immune cell, for example, may give its nutrients or things that it might consider wastes to the cancer cells. The cancer cells can use those. And so there's this sort of shared symbiotic relationship that exists among the cells in the tumor who all have the same desired outcome, which is to grow. Now, reciprocally, there's also competition that exists because our immune system, one of its jobs is to surveil the environment and look for things that are unnatural, like tumors, and to target and kill those. Well, immune cells, like cancer cells, in order to do their job, also need food. And the cancer recognizes this, and it deprives the immune cells, the anti-tumor immune cells, of those nutrients through direct metabolic competition. So if you're a T cell, you need to make more T cells. You need nutrients to do that. The cancer eats up all those nutrients so that they're not available, and the T cells are less effective. And this is now recognized as a major mechanism of immune suppression in cancer. And as we go through, and think about these concepts of rewired metabolism, there's always an eye on the goal line, which is, can we think about new drug targets and new therapies? All right, so let's jump into a little more specifics here. In pancreatic cancer, what we've learned about their metabolism can be simplified in some ways into sort of a singular statement, in that the way they wire their metabolism is to become experts at scavenging and recycling. So one of this, from a scavenging perspective, I've sort of talked a little bit about this. What I've shown here is a simplified schematic where there's communication among the different cell types in a tumor. Some of those are symbiotic to promote growth. Some of those are competitive, so it's to keep the immune system away. So this sort of interacting. The other bit we've become to appreciate is that the cancer cells become really good at eating whatever is around them and recycling the things that are inside of them. And that's the point I'm gonna stick on a little more today. So that's the heart of today's seminar. Now, these two pathways that I'm going to talk about, autophagy and macropinocytosis, are two recycling and scavenging pathways that converge on the lysosome. The lysosome is this organelle that has, I guess, long been given the dirty name of being the cell's trash can. And that's where all the waste ends up. And once the waste is in there, there are a variety of mechanisms that can occur in order to break down whatever comes in into subsequent building blocks like amino acids and lipids and sugars, and those can be fed back to the cell. So it converges on the lysosome. And what we know, I'm going to go through this relatively quickly. What we know about autophagy, which literally means self-eating, is that under conditions of nutrient stress, so if you think of a normal cell and you think of a healthy person who didn't have access to, maybe you're fasting, the cells in your body will start undergoing autophagy. They don't have access, the body has less nutrients around, and so the cells will turn on self-catabolism to maintain energetic homeostasis. This is a built-in normal mechanism for maintaining metabolic homeostasis. Well, in pancreatic cancer, what we appreciate is that even when the cells have nutrients, so we can take the cells out of the tumor, we can grow them in a dish, they keep this pathway on all the time. There've been a lot of questions about what is the relevant output of this pathway, and I'll talk a little bit more about that. I don't want to belabor this slide terribly, given the composition of the audience, depending, sometimes if I'm talking to people who study pancreatic cancer metabolism, I really get into the weeds here. And there's also an appreciation that under stress, the already high basal levels of autophagy go up even higher. And so there's been a lot of thought and preclinical activity around targeting this pathway for pancreatic cancer. The other side of the same coin is this pathway called macropinocytosis. This is also known as cell drinking. So it's another name, cell drinking. And in essence, what a cell will do, as somewhat illustrated here, and I'll show you if you're looking at my screen, it'll put out an arm and it'll bring in some extracellular space, non-specifically, drink it. Bring that in through a macropinosomes, and these processes all converge on the acidic lysosome, whose job it is to break down those whatever macromolecules are there. So it stands to reason then that a pancreatic cancer cell will have higher lysosomal activity, and indeed, several labs have illustrated that lysosomal biosynthesis turnover is upregulated through a transcriptional program that's hardwired into pancreatic cancer cells. Okay, all that to say, that's the background. If we had a way to target the lysosome, this would seemingly be a great way to treat pancreatic cancer. But the compounds that we have to do this, so some of them like baflomycin are great tool compounds to study this in vitro. And then we have another class of compounds called, if you're following my laser pointer here, chloroquine, hydroxychloroquine. And these are medicines that are used to prophylactically treat malaria. They work well are able to access the blood, but they do a poor job penetrating tissue. So just based on drug properties. And clinical trials have been run with these molecules, and they have been wholly unsatisfying. No movement in patient overall survival, and then worse, they've not, if you look at molecular markers of lysosomal inhibition in tissues, in pancreatic tissues, we don't really see pharmacodynamic markers of activity. So it's not that the idea doesn't work, it's that the drugs don't work. And so we still have this really interesting idea that is out there to test. So that's where this story picks up. We have a new target that goes after the lysosome. It's called PIK5. PIK5 is a lipid kinase. It phosphorylates PI3P to make PI3-5P2. And to make a long story short, it is involved in lysosomal homeostasis, so lysosomal trafficking turnover. We have some new medicines to target PIK5, and these I'll talk about throughout the talk today. So, and I'm going to use some acronyms here. Hopefully you can follow along. One is called apilimod. Apilimod is a great tool compound. We use it for all of our molecular studies, but it has poor pharmacokinetic, pharmacodynamic properties as well. So couldn't test this hypothesis, but what we have landed on recently is a multikinase inhibitor called ESK981 with really promising activity in pancreatic and other cancers. And it does so by targeting PIK5, that's the relevant molecular target. And in addition to this compound, we've also made degrader molecules. So for people who are interested in study those sort of things, these are molecules that simultaneously will bind to target and then recruit the proteasomal machinery to literally degrade the target. So these are called degraders. They're also called PROTACs. Depending on what flavor you're looking at, there are also things like molecular glues. So those are some terminologies thrown around, and we have built those on the scaffold of ESK981. And I should mention we are doing all of this work with the Roel Chennaiyan's group, and I'll follow on with a little more of that in a second. So PIK5, that's our target. Our clinical candidate agent, ESK981, is in clinical trials at the University of Michigan in the United States. But before I jump into the data, I like to start with a high-level summary overview, and that serves two purposes. One, it lets me acknowledge the people who actually did the work. Not, I get to take some of the credit for having participated, but really it's Caleb, an MSTP student in my lab and Roel Chennaiyan's lab, and Yuan Chow, who was a postdoc and has moved into a junior faculty position at Michigan, who did the lion's share of the work here. And what they found, and then the second thing is I like to give you a summary ahead of time so that it's easier to follow along and track, what's Costas's logic as he's moving through this. And so that summary is as follows. What we've identified is that in pancreatic cancer and PDAC, pancreatic ductal adenocarcinoma, lipid metabolism, lipid homeostasis is maintained through two parallel arms, put simply. First is the de novo biosynthesis, so building lipids from scratch. And this is a molecular program regulated by the KRAS MAP kinase signaling pathway. KRAS is an oncogene. It is mutated in nearly every pancreatic cancer in human beings, and it is a major driver. And if we can shut it down, you have major impacts on tumor biology. And another interesting and important thing about KRAS is when we started this story, and I mean, as little as say 10 years ago, KRAS was considered an undruggable target. And that was a bummer for the field because I just told you almost all pancreatic cancers have KRAS mutations. So if we could target this transforming signature oncogene, it'd be great. And then over the course of the last 5 years, and even during the last couple of years, these inhibitors, new clinical grade inhibitors, are in clinical trials for pancreatic cancer. So a means to target this pathway is now available. Preclinically, it's not clinically approved, but it's really exciting times in pancreatic cancer. And one other point, I'm getting a little ahead of myself. What has been observed in clinical trials, and we know from preclinical models, and we know from other targeted therapies, KRAS inhibitors will work really well for a finite amount of time in a patient, and then the tumors develop resistance. We've seen it with every targeted therapy that we've done in oncology, so this is a known quantity, and what we and others now need to do is get ahead of it. So how can we find combination therapies that work well with the KRAS inhibitors? And that's another major underlying theme and message of this story today. Okay, so let me go back. Lipid homeostasis is regulated by two parallel pathways in pancreatic cancer. The making of it through RAS, and then the recycling of it through lysosomal processes. If we inhibit PIK5, we inhibit the lysosome, the cell compensates by ramping up de novo biosynthesis. If reciprocally we inhibit RAS, the cell compensates by ramping up lysosomal recycling of lipids. And what we've identified is that this creates a synthetic lethal relationship where if both pathways are targeted simultaneously, then the tumor is susceptible and it's really aggravated. And we see a lot of cell death, and we see really remarkable responses in our models. One of the most striking observations for me in having worked on this now is that the normal cell, the normal body, the normal organism tolerates the combination so well, while there is a cancer-specific vulnerability. I mean, I suppose that's what we are looking for as cancer biologists, as oncologists, is how do we identify cancer-specific features? But it's still, it's remarkable and satisfying when you do find one of these, real tumor specificity. Okay, so I'm already feeling a little long-winded. I'm going to jump ahead and briefly touch on this slide. This work here on this slide is published. And what we demonstrate is that this PIK5 inhibitor, this is the first time it's been applied in pancreatic cancer. And so we needed to make sure that it was in fact inhibiting the lysosome. And if you are to look very closely at your screen, you can see that in PIK5 inhibitor treated cells, they get these massive vacuolization phenotypes, and that's a reflection of the inability to turn over the lysosome. So those are lysosomes that are literally filling up with fluid and can't turn over. And some cells will go on and die from that, and some cells will just persist for a long time until either they figure out a workaround or we kill them with a combination agent. And these are some metrics to illustrate that we inhibit lysosomal and autophagic turnover. But in the interest of time, I don't want to belabor that. For people who are really interested in that content, we can talk about it during questions, or we can I can point you to the paper. So this seemed like a cool inhibitor. If you'll remember from my background slides, targeting the lysosome, there's a lot of really great rationale to do that. And what I also told you is that we didn't have good medicines to do that previously. So ESK-981, or labeled here ESK, is different from all of the other ones in that it can be delivered daily. We do it in the mouse by IP, but in people it's taken as a pill. And it has really great penetrance so it gets into the bloodstream, it remains in the bloodstream, it has a good half-life, and it can get into tissue, and most importantly, it can engage target in tissue, and we have a bunch of data to illustrate that in both mice and in people. I won't belabor that, just trust me on saying that it is a good medicine. Okay, but is it a good drug? Does it treat tumors? So this is the first example. This is, if you are a cancer biologist, you'll know that this is not a fantastic model. These are cancer cells that we grow on plastic that we then take and put under the flank or in the subcutaneous space of an immune compromised animal. So it's like it's the most artificial in vivo model, but in this model system, it works really well. So ESK will flatline these tumors. You see a lot of them will shrink from the initial size when we start treatment. That's compelling. I will tell you, however, as soon as we remove treatment, the tumors grow back. All right, so we tried some more complicated models, similar stories. This is an immune competent model. This is a human patient-derived model. It's because of being human, it is immune compromised. So each of these these models have varying strengths and weaknesses. And in all of these models, we see that we have promising single agent activity, but non-curative. We also asked ourselves what's happening to the tumors, and we know that this is a short-term treatment, that there are a number of the tumor cells that are dying by apoptosis. So single agent treatment looks promising, but it's not good enough. And I like to tell the trainees that I work with, if you can't cure a mouse, you're not going to cure a person. So good, but not good enough. So how do we make this better? So that is the challenge that Caleb took to. He said, how can we identify synthetic lethal relationships with PIK5 inhibition? And he used two parallel strategies. The first was transcriptome profiling. The second was synthetic lethal CRISPR screening. In the first, what we did was to treat, here, this is a mouse cell line treated with two different PIK5 inhibitors, and then we looked at the gene set enrichment, and these are distinct pharmacophores, so they have the same target, but they have different chemical structures. And what we saw was a remarkable convergence from the gene set enrichment analysis, which gave us confidence that that PIK5 was the relevant target. And what we saw was this increase in transcripts related to the making of lipids. You'll remember from my earlier summary slide. Okay, so no surprises here. When we inhibit the lysosome, the cells say, we still need fats. We're just going to make them instead of scavenging them. What we identified is, so these are transcripts. They're downstream of a master regulator of lipid biosynthesis called SREBP. I won't belabor this too much, but for people who are interested in lipid biosynthesis, this is interesting stuff. From a premature to a mature processed form, when you use any one of three PIK5 inhibitors, it goes, you can see that this spliced version, which is the active transcription factor, accumulates. So that's reflective. And I will also say that now I'm getting a little deeper into the weeds, but we also demonstrated that distinct lysosomal inhibitors like chloroquine and baflinomycin have the same phenotype. So this wasn't unique to PIK5, but it's this feature of enhancing lipid biosynthesis in pancreatic cancer was a consequence of inhibiting the lysosome, not necessarily PIK5, but more big picture inhibiting the lysosome. So from our paper, this was a compelling molecular observation that hadn't been reported. It's for people who are interested in this, we think it's super fascinating and are studying it in greater depth. But so I'll just mention that there. We looked at transcript. Those were up. We looked at protein. So that's the SRBP transcription factor. Those are up. And then we also did lipid profiling and isotope tracing. And I won't belabor all of this to say, but it's not just the transcripts that are going up, but it's literally the lipids are being made, and we demonstrated that. I'm going to skip this slide in the interest of time and jump to this next one. So I told you in parallel, Caleb was running CRISPR screens and the transcriptome profiling. So I showed you transcriptome profiling. Here's the CRISPR screen. The way that you can visualize this data, well, let me describe the screen first. So what Caleb did was to take pancreatic cancer cells, transduce them with a library that has guide RNA, so CRISPR guides, 10 per gene for this is a metabolome targeted library. So we were looking at about 5,000 genes, 10 guides per gene. You transduce a population of pancreatic cancer cells with that library. Then you treat them with drug, grow for 2 weeks, and look for you sequence at the beginning, you sequence at the end. And at the end, when you sequence, you look for genes that fall out. So they're de-enriched. And these are things that are synthetic lethal. And you can also look for things that are enriched, and those are genes that would promote growth with PIK5 inhibition. And these are ones that make growth worse with PIK5 inhibition. So what are these? I've laid them out on this schematic to your right. You don't need to be an expert to hopefully appreciate that the top 30 genes that we found are shown, several among them are shown in red and purple, and they populate onto a single metabolic pathway, which is the lipid biosynthesis. I guess maybe it's not a single, you could call it two metabolic pathways. The first is the de novo fatty acid and lipid biosynthesis, and the second is de novo sterol biosynthesis. So this is how you make, among other things, cholesterol. And I mean, it was an extremely satisfying amount of populating of this pathway, really illustrating to us that, when you pull one or two genes, maybe it's an artifact, but when you pull pathways, it seems like there's really clear biology. And I will say, as a brief digression, having run hundreds of CRISPR screens, this is absolutely the most satisfying one I've ever seen. So this is like really a robust phenomenon. So what Caleb then did was to test, when you get data out of a screen, that's great, but you need to independently validate it, and even better, independently validate it with an orthogonal strategy so that you're not being tripped up by some weirdness or artifact. So that's what Caleb did. But before I talk about that, I'll just quickly show you on the left-hand side, if you're making fats from scratch, cells do that from, among other things, glucose. And these are the steps in the pathway. What I've shown in red are a few of the enzymes that were synthetic lethal in our CRISPR screen and that Caleb assayed. So the first is ACC1. There's a drug against ACC1 called ND646. If you try either that drug at a concentration that doesn't really impact pancreatic cancer growth, shown in red, or apilimod, shown in green, either one of those at those doses, a single agent, have really no meaningful effect on pancreatic cancer proliferation, but the combination is growth inhibitory. Okay, that's satisfying. That reflects what we saw in the CRISPR screen. And if you march down this pathway, the same was true of FASN. Here we used CRISPR guides. In essence, same phenotype. And then here's a third example, just really to drive this home, that if we target SPTLC, same story. Single agent, no meaningful effect. The combinations, in this case, were killing pancreatic cancer cells. So that's well and good. And now we have a bunch of targets we can go in vivo with to examine synthetic lethality, right? We have PIK5 plus, let's block de novo lipid biosynthesis. So that seemed like a good strategy. And as we were further interrogating this, I'm going to distill like 10 years of my life into a single point. I've been working on the regulation of metabolism by KRAS. And so we made the hypothesis, the logical hypothesis, that maybe all of this pathway was being regulated by KRAS. There were a couple other indications that was the point. And indeed, KRAS does regulate this pathway. In the interest of time, I'm just going to ask that you take my word for it. We do show, using up and down assays, that when you inhibit KRAS, these genes, FASN on the top, ACC1, this is the gene name for ACC1. When you inhibit KRAS, those go down. If in reciprocal pathway you activate KRAS, those genes get turned on. So we can block KRAS and turn them off, or we can turn on KRAS and turn them on. And I also want to remind you that KRAS does a whole lot for pancreatic biology. So We knew that if we took advantage of these KRAS inhibitors, we'd be hitting lipid biosynthesis, but we'd also be hitting other features of pancreatic cancer. And we know that these KRAS inhibitors are making their mark in the clinical trial space, and my instincts are that this will be standard of care in short order. So being able to capitalize on the KRAS inhibitors, there was also a compelling reason to pursue it from that perspective. I will also say that at the end of the day, these KRAS inhibitors plus PIK5 inhibitors proved to be the most effective combination strategy in vivo relative to targeting any singular arm of de novo lipid biosynthesis. Okay, so in the last 5 minutes, what I'm going to do is take you through some in vivo data. And those are data where we pursued the following hypothesis. If you inhibit PIK5, you turn on de novo synthesis. If you inhibit de novo synthesis, turn on the lysosome. The combination should be synthetic lethal according to our in vitro studies. So we did that in at least 3 model systems of increasing complexity, as I'll present them. The first is a human cell line derived xenograft. So we implant those cells into immune compromised mice. These are human cells, human cancer cells, and we give them single arm treatment of either, so vehicles in blue, the MAP kinase, KRAS MAP kinase inhibitor is in red, and the PIK5 inhibitor is in green. The combination's in purple. You can see that the combination shrinks tumors meaningfully, and at endpoint, all of the animals on combination treatment, their tumors had not doubled. Super exciting. This is what one could assume is a cure, but I will tell you that if we take these animals off treatment, the tumors will grow back. And if we look at the pancreas at endpoint, there is still some residual tumor there, consistent with the fact that the tumors grow back. So, like I said, curing, if you can't cure a mouse, you're not going to cure a human. But this was compelling. This was compelling preliminary data. We're shrinking tumors meaningfully. So the next thing we did was to examine this combination in a more complex model. That is, we take murine mouse pancreas cancer cells that are genetically identical to mice, and we implant those cells into the pancreas of those mice. So what you have in this context is a mouse with an intact immune system, and we put the cells into the pancreas, which is the relevant microenvironment for the tumor to grow. I'll try to move through this relatively quickly. Single agents had modest activity. The combination literally cured the mice. So when we take these mice off of treatment, there is no tumor regrowing in 80% of the mice. 20% or so would have some regrowth. But what we are doing here, this is one of the most complex models for this disease. The tumors are fully, frankly cured. Super exciting. What it also illustrates to us, what's different between this model and the previous, these mice have an immune system. So maybe there is some additional immune activity that's giving us that added benefit to push it over the edge. This is the last model I'll show you. This is the most complex model of pancreatic cancer that the field has. And to my most recent reading of the literature, no one has ever cured this model, which makes it a great surrogate for human disease, which we also don't cure. So there's a high bar. Single agents are shown in red and green, combinations in purple. This work was done with Jen Morton in Scotland. And what Jen does is to use ultrasound. I didn't even describe the model. So the model is an endogenous tumor model. It's a genetically engineered mice. So we engineer these mice to express an oncogene KRAS and a tumor suppressor P53 in the pancreas. So we use a pancreas-specific CRE system to drive expression of this oncogene and this tumor suppressor in the pancreas. This model is called KPC for KRAS P53, and then the CRE. And the animals will develop spontaneous pancreatic cancer. It looks histologically like human disease. It can't be treated like human disease. What Jen does is after about 2 months, every 3 days, she does ultrasound on these mice and waits until she's able to see tumors that are 50 cubic millimeters, and that's when she starts therapy. Okay, so just like a human patient would come into their oncologist. The oncologist would say, we see a mass in your tumor, let's start treatment. That's what these mice get. They have a naturally arising tumor. We wait until it's observable, and then we start treatment. And then those tumors can be followed by volume using by ultrasound, and you can see that in the single agent arms, the tumors grow, like in the vehicle. In the combination, the tumors in the pancreas didn't grow, and if anything, they shrunk quite a bit. This is the best response we've ever seen in this model, and I would love to say that we cured these animals, but you can see from the Kaplan-Meier curve, while we move it about fourfold, we don't cure any of the mice. The mice don't look like they have tumors in their pancreas, but they're dying of something else. And we're in the process of figuring out what that is, hopefully, and identifying additional strategies. Maybe that's turning on the immune system to further to lead to more lasting remissions and cures. So that's the last piece of data. I had a couple slides on how we activate and what we think about the immune system, some unpublished work. I'm going to skip over those in the interest of time. And if people are really excited about that, I'd love to go into it during the question and answer session, but I want to leave time for discussion and let the audience members dictate which direction we go. I'll just conclude by saying we demonstrated that in pancreatic cancer, lipid homeostasis is regulated by, put simply, these two parallel pathways. Combination inhibition is synthetic lethal. That we are now actively moving these ideas into the clinic. We're in conversation with several. There's something like 40 different companies that have KRAS inhibitors. I mean, it's a crazy busy landscape right now. 40, literally, have KRAS inhibitors. So we're talking to some of them about testing KRAS inhibitors with our PIK5 inhibitor in patients. I'm not following exactly in the order here, but we want to test that with ESK-981, so not KRAS alone. While that is important, we want to do the combination. And ESK-981 is a clinical candidate. We have it in a phase 2 trial at Michigan as a single agent. And I'll just say the last point there, that while KRAS inhibitors are undoubtedly going to change the landscape of the treatment paradigm for pancreatic cancer, we already are observing resistance in the patients in clinical trials. After about 6 to 8 months, upwards of a year, those tumors figure out ways around. We see the same thing in our preclinical models. So we need to identify combinations. We think this is one such combination, a really compelling lead. Maybe it needs some additional immune stimulatory modulator. But yeah, so that's that. I thank the group. This is my research team at the University of Michigan. This work was led chiefly by Caleb and Yuan. We had really tremendous support. It's through a very active collaboration with the Rule Chennaiyan's group at the University of Michigan. This is a rules group. He's an HHMI investigator, for those of you who know what that is. He's got a monster group. He is a world leader in genomic sequencing, and among other things. We have a number of other collaborators to thank. Great team at the University of Michigan with Marina and Yatrick and Tim and so forth. I mentioned Jen's lab helped us out at the Beatson. Always a pleasure to work with Jen. She's fantastic. And I'll close by saying we just started a pancreatic cancer center at Michigan that was endowed by a $50 million gift from the Rogel family. So there's a lot of exciting activity in pancreatic cancer happening. We are recruiting literally at all levels. We're looking at PhD applications start December 1st. That's when they're due. Several of our labs are looking for postdocs right now. For any graduate students on the line looking to postdoc, things in America are unusual right now in science, but it's a storm we think we can weather. And with this sort of support and the great team around us, it's been okay so far. And I'll also say that postdocs out there working in this space, we have an open faculty call as well. So we're looking for faculty in GI and in pancreatic cancer. All right. Thanks for bearing with me on a Zoom call. I know that these aren't always the most fun. Hopefully I made it sufficiently engaging, and I'd love to interact now. Thanks so much.
Costa, thank you for the wonderful and very insightful talk. I think I can talk for everybody. It was I mean, very hopeful to see that you had some kind of treatment in the experimental models for pancreatic cancer, especially considering that it's untreatable in the humans. So that's really interesting. And it's good to hear that there is hope. Things are going to be better in terms of research. I'm sure there will be at some point. You always have to go down before you go up, right? So before I don't see any questions coming up yet, so I'm just going to take the advantage of, and I guess the first question that I have for you is from the in vivo results that you presented. So because I'm not familiar with the field, what's the exact difference between the allograft model and the second in vivo model, the KPC that you showed, and that kind of justifies having treatment in one and no treatment in the second one?
Yeah, no cures in the second one. Yeah, really insightful. Thanks so much, Maria. Really appreciate the opportunity to present. And thanks again, everyone, for their attention. So the first model is, like you said, an allograft. So we take cell lines, we've developed cell lines from the genetically engineered model, and we grow those in culture, and we can manipulate them. We can do whatever we want with them, study them in culture and so forth. Those can be expanded, and we can make a cohort of 40 mice. So we get black 6 mice, that's the genetic background. We grow enough of those cells to implant 50,000 into each of 40 mice. And then we randomize those 40 mice and we say, you're going to get vehicle, you're going to get treatment A, you're going to get treatment B, you're going to get combination. And we put 10 on each arm. And then that's really standardized. They all got the same cancer cells at the same time. They're going to grow tumors in their pancreas. And we follow them for 30 days. So it's a super standardized process and we can do it. There's an immune system and it's in the pancreas. So those are the pros. The con is that we dump a bunch of cells into the pancreas. And so that's not how cancer presents. So that's where the second model comes in, the more complex one. That one is called, it's a genetically engineered model. People use the acronym GEM for GEM, genetically engineered mouse. And that one is another name for it, in case you're following all the different terminology, it's called an autochthonous model. In that model, we don't touch the mouse pancreas at all. We have hardwired into their genome mutant KRAS expression that can be turned on as their pancreas is developing. So it's using Crelox technology. And so as the mouse is born and during the first couple weeks of life, KRAS and mutant P53 get turned on in that mouse's pancreas. And so because it's now that pancreas is hardwired with an oncogene and a tumor suppressor, that mouse is going to get pancreatic cancer. And that pancreatic cancer, it is coming on in its natural environment. So 1 cell turns into 2 cell, turns into 4 cell. And what's happening there is it's talking to the immune system in the mouse as it's developing. It's changing the architecture of the pancreas. And this is exactly how, we don't engineer humans to have cancer, but as mutations arise in a human, this is exactly how the cancer is evolving in the person, right? Mutations occur in a single cell, and those single cells go on to form a tumor. And so what this gives us is the most representative tumor model of human disease that we can make. And like I said, no one can cure it. It's super treatment refractory. It has a short latency. It kills all the mice. And so it's the bar at which we set to say, if we can cure this mice, we have something that's interesting to take to people.
Okay, yeah, I mean, it's a more natural condition, but it was interesting that you mentioned that you don't know the cause that these animals are dying from. So maybe it's a population of unknown cells, maybe, or some other component of the microenvironment that it interacts with the original tumor, perhaps. You don't know yet.
So that's the big question, and I'll tell you where I love where your head's going. I mean, it's great speculation. I'll give you some further speculation from us. This model will metastasize,
Okay, that
and
was neat.
so we didn't look at the Mets, but we know that it is metting, and we didn't look at control of mets. These are typically micromets, so it might be impacting organ function. We know that these mice also get sick, just like humans get sick. They
Okay.
have muscle wasting, they have body wasting, they have immune suppression, and so they are susceptible to infections and things like that. So it might just be because they're sick that they're
Yeah.
dying. That's part of it. So we didn't cure all the cancer, and there's enough cancer to still cause that kind of trouble. We know that these animals are sick, and maybe we don't see toxicities with the drugs in healthy mice, but
Mm-hmm.
in these mice that are sick, maybe the drugs do add up to becoming toxic under those circumstances in
Yeah.
some way that we don't yet appreciate. It's not clear. We're definitely very interested, and I mean, it's keen observation there.
Yeah. So, I mean, that was another. So, I actually had a relative of mine that died of pancreatic cancer. And I mean, uh, she did it was a very stressful, she had rheumatoid arthritis and all that, and she had other conditions that burdened her body over the years, and ended up developing the cancer. So what's the genetic background for this type of cancer, and what's the epigenetic or environmental component globally? I guess if I can take it in a more summarized question.
Yeah, yeah, fantastic question. I'm sorry to hear about your relative. Um, we can account for about 10% of pancreatic cancer based on genetics. So
Okay.
people who have BRCA mutations, LKB1 mutations, Lynch syndrome, there's a subset of individuals who have germline familial mutations, and if we identify those individuals before they have pancreatic cancer, we have them in a more an annual screening program.
Okay.
So they'll get ultrasounds um, regularly, whereas we don't do that for the general population because it's not an effective use of time or money. That's about 10%. So what about the other 90%? There are a couple of risk factors, which include inflammation in the pancreas, pancreatitis, smoking, alcohol consumption. Um, this is one of the obesity-associated cancers, uh, lack of insulin control. But all of these are, to be honest with you, really modest contributors. They increase an individual's likelihood uh, so this is also a disease of age. So most patients will present with pancreatic cancer after the fifth decade. Not universally true, but most. And then the likelihood, if you're a smoker and a drinker, maybe you increase your likelihood 2 or 3 fold, which is not nothing, but it's also not the story. So um, we're not sure.
Okay, so it's pretty mysterious, I would say, still the whole situation around it. And I was wondering, I mean, you started the topic of your talk was lipids, right? So uh, one part of the question is what actually happens to the lipidome? Like what are the changes that you see like histologically in the pancreas per se when you give treatment, and how does that, have you looked at the changes in the two models, especially in the one that's more physiologically relevant in the lipid, I don't know, microenvironment, or the interactions with the microenvironment of the lipid component versus the immune component also? It was a pretty complex question, so.
Yeah, no, I think I got the heart of it. Sort of two related questions. What's happening, what changes are in the lipidome, and uh, with individual treatment, we see that de novo goes way up, and with if if you use a PIK5 inhibitor, de novo lipid biosynthesis goes up. We see an enrichment in sphingolipids and in ceramides. Don't really know what to do with that information yet.
Okay.
But your second question, I think it may be a more, for me, compelling one, and in particular relates to some immune effects that we're starting to see. So what's happening to all of the other cells? I started by talking about tumor microenvironment. What happens to all the other cells when you inhibit these things? How's it
Yeah.
rewiring? And to be honest with you, we are just starting to dig into that. I have a graduate student who's working on the fibroblasts. I have a graduate student who's working on the cancer cells. I have a graduate student who's working on the immune cells. We can tell you for sure that there's a lot of action. We know that with single cell sequencing, we see populations moving all around. We don't know if that's which is cause, which is consequence, and we're digging into that now. I think it's a really exciting area. So yeah, great questions.
Yeah, I guess the microenvironment is the goal. So we actually have a question from Vasilis Georgiou. Thank you for the amazing presentation, which it was an amazing presentation. I have a question regarding the tumor size threshold you use to initiate treatment. Is this threshold based on the equivalent range typically diagnosed in humans? I am wondering whether starting the treatment earlier might lead to different results, especially since you observed a significant reduction. I ask because AI-based diagnostic tools are now being developed to detect tumors much earlier, although they're still in early stages, and it will be interesting to see how earlier intervention could impact your findings.
Yeah, thanks, Vasily, for the question. Excellent one. I brought it up here too. I'm looking it over. So the 50 cubic millimeter that we choose to detect by ultrasound, it's the standard in the field. It is sort of the smallest that an ultrasound can reliably detect. That one of these mouse ultrasounds. So you literally you take a mouse and you're doing an ultrasound on them. Like think about when a woman is pregnant, she goes to her doctor and you put the gel on and you rub the probe and you can see that the baby moving. This is the same sort of technology that's used, right? So that's when we can reliably diagnose a mouse with pancreatic cancer with assurance. So if we started earlier, we would be guessing before 50 cubic millimeters. And in these mice, there is varying onset. It can vary by as much as several months. So you might start a mouse on treatment and then it doesn't get a tumor for 6 months. And so you don't know if that's because you cured it or because it just wasn't going to get it. So that's I think, technically a tricky one. And it's something I would love to do more of. I'll just say that the ability to run a mouse hospital like Jen Morton does, it's not a trivial or inexpensive. And so we have limited access to tumors that we can treat there. But I love where your question, the heart of the question. So I answered some of the technical mumbo jumbo, which is if we're able to detect these earlier or let's talk about the mouse versus the human. In humans, sometimes a patient will have pancreatic cancer. We'll know they'll have, we can see metastatic pancreatic cancer in that patient, but we don't see primary because the primary remains so small and below our detection threshold. So that's a wild circumstance that we can say this patient has metastatic disease, but we can't find the primary because it's so small. And that's another feature of pancreatic cancer. It metastasizes very early in its evolutionary history. And so if there were methods to detect earlier, that would be awesome. I would love to see AI come in and help us out with this. I'll just introduce, I don't know if I'm answering the question or just blabbing, but I'll introduce one other complication. The pancreas is like right smack dab in the middle of our bodies. So like middle of your chest, both from the front and from the side. It is a difficult organ to access, and unlike accessible areas, skin cancer, breast cancer, even colon cancer, we have easy ways to get to these tumors and be able to look at them and then apply strategies to start diagnosing. A routine ability to screen a person's pancreas still remains a challenge.
Yeah, that's the same problem with the brain, right? The tumors in the brain are very difficult to access because of the sensitivity and difficulty to, depending on the location, especially. So along those lines, Do you think that what you see in the pancreas, like with the lipid metabolism and the targets that you detected, could be applicable to other kinds of cancers, especially
Oh,
because we
yeah.
know that, yeah.
I love that question too. So, yes. And what we've found, so this is work that Arul Chanayan's lab, our collaborator on this, is doing more of. What he's demonstrated is that, and what we've demonstrated, when you inhibit PIK5, so when you block the lysosome, the cell becomes dependent on making new fat. But all cells don't do that the same way. So we have two other studies that are at some stage of the publication process where in one case, we know that the way that they make the fat is downstream of mTOR.
Mm.
And so instead of inhibiting KRAS in that disease, we use PIK5 plus mTOR inhibitors, and we see similar results. So you just need to know what is the genetic driver. In another disease that we're working on, we don't know what is the major driver of lipids. And so in that case, so this is prostate cancer. Well, we target FASN. We target lipid biosynthesis directly as opposed to any singular pathway driver. But the combination works great. Sort of this paradigm holds. You just need to know what's making the fat in that context. In the pancreas, it's all about KRAS and MAP kinase.
And Dr. Vasily says, thank you for the answer. And
My pleasure, Vasily. Thank you.
and just so I don't want to take any more of your time, but I have a question because I was on a study that we looked at epigenetic regulation of lipid synthesis in the liver and in relation to liver cancer and other liver-related diseases. So we
Oh, yeah,
saw
okay.
that NA40, that acetylates histone 2 and 4, was implicated in the metabolism. And that was in mouse cells and also in Drosophila, we saw the similar. So I was wondering if you looked at like any epigenetic components, especially as you know, this is an age-linked disease, and you know, aging can have a great interaction with the epigenetic component. So, have you looked at that? Are you
I,
looking at
I'm
it?
I'm I'm going to give you the most unsatisfying answer in that no, we
No.
haven't we haven't looked. That's really great thoughtful feedback and comparing the liver. No, we haven't looked at anything related to the epigenome
Okay.
and its control. We've sort of looked at the transcription factors that regulate
Mm-hmm.
this process, but not the epigenetic state of the genes or anything in that regard.
I mean, it's a lot to look at because you have so many factors, you know. We can't forget that we focus on specific pathways, but in the end, you have to do with the system. The greater challenge is putting everything together in terms of how it functions as a system. So these are excellent results. So yeah, way to go. And good luck
Thanks.
with the clinical translation, because you said that you are working on that, right?
Yes, very excited about the possibility. I'm hopeful
Okay.
we can make some progress there.
Yeah, so good luck, and you know, keep us posted on the progress.
Sounds great. Will do.
Um, Thank you very much, Costam. I don't see any other questions from our audience. I don't want to take any more of your time, although I'm very tempted to. I've noted everything down. But it was a wonderful talk, very insightful and hopeful for cancer and how research is going in that field. Thank you very much again for initiating our 10th season. And
Oh, yeah, what a pleasure.
yeah, the very best for your day.
Thank you so much. And if anyone has any other questions, I'm happy to address by email. So thank you. Really appreciate the opportunity, Maria. It's been a pleasure.
Thank you. It was my pleasure. Kalimera.
Yeah.
See you. Ευχαριστούμε όλοι.
Yes,
Bye.
yes.
Thank you.
Bye-bye.
Bye.